recombinant DNA technology and the other molecular base of cloning technology. What is cloning technology? Generation of many copies of DNA, for example, recombinant DNA by replication in a host. Developed in 1970s, can generate unlimited supply of gene copies. Many applications, genetic genetic mapping, in DNA sequencing, in mutation studies, in transformation, in genetic engineering. We form recombinant DNA libraries, restriction enzyme type analysis of gene transcription, what is being expressed, polymerase chain reaction PCR, DNA sequencing to be included with other topics. Goal is to generate large amount of pure DNA. Goal is to generate large amount of pure DNA that tabulated and studied. DNA is cloned by the following steps. Isolate DNA from organism, extract DNA, cut DNA with the restriction enzyme to a desired size, splice or ligate each copies of DNA into a cloning vector to create a recombinant DNA molecule. Cloning vector. What's cloning vector? Cloning vector, artificial DNA molecule capable of replicating in a host organism. For example, bacteria. Transform recombinant DNA cloning vector plus DNA fragment into a host that will replicate and transfer copies to protein. There is an overview. Step 1. Isolate the whole genome DNA from the organism. How can we do this? This step, DNA extraction easily performed using a detergent or sodium chloride or proteinase kinase or ethanol or water. Break up cell membrane and organelles by detergent. Sodium chloride lyses cells and binds the DNA strand together. Proteinase kinase to digest protein bound to DNA. Ethanol to precipitate and wash DNA. Water to resuspend and store DNA. Storage. DNA can be stored short term at room temperature, but it's best stored long term at -80 centigrade or in liquid nitrogen. Ideal storage. The ideal storage, large uncut DNA fragment. Suboptimal storage condition, many small DNA fragments. If we put it or store it in suboptimal conditions, we get many small DNA fragments. Average size of DNA fragment is important for application. Involve large regions of DNA sequence, less important for application. Involve short region of the DNA sequence. The restriction enzyme. Restriction enzyme. What is restriction enzyme? We cut the DNA with restriction enzyme. Restriction enzymes. Restriction enzyme recognize specific base pair. Recognize specific base pair. Restriction enzyme recognize specific base pair sequences in DNA called restriction sites. Restriction sites, and they cleave the DNA by hybridization or hybridizing the phosphodiester bond. Cut occur between 3 carbon of the first nucleotide and the phosphate of the next nucleotide. Here the site of cutting. Restriction fragment in the half 5 phosphate and 3 hydroxyl. Cut the DNA with restriction enzyme. Most restriction enzyme occur naturally in bacteria. Protect bacteria against the virus by cutting up viral DNA. It one of the methods of protection or uh, self-protection from the bacteria against the bacteriophage. It cut the DNA of the bacteriophage to defend itself. Bacteria protect their DNA by modifying possible restriction site methylation. Uh, protect their DNA by modifying possible restriction site methylation. Change the DNA to protect its DNA. More than 400 restriction enzymes have been isolated and this increased every day. Names typically being with the three etylized letters, the bacteria and the number E. coli R1, R1 and 3 BAM H3. This is echo R1 from E. coli, and 3 Haemophilus influenzae, BAM HI Bacillus amoebicoefficient H. Many restriction sites are palindromes of 4, 6, or 8 base pair. Short restriction site sequence occur more frequently in the genome than longer restriction site sequence. The short restriction sites occur more frequently in the genome than long restriction sites. For example, echo RI sequence is symmetrical about the center point, the point of cleavage here, this point of cleavage. Digest with echo RY here, the product, the double strand RNA become like this. Cut the DNA with restriction enzyme. Some restriction enzyme produce blunt ends where other products stick overhanging stacked ends. Sticky ends or other overstacked ends. Stick ends are useful for DNA cloning. Overhanging or sticky ends. Here a blunt ends, overhanging sticky ends, overhanging sticky ends, info and up, here down. Sticky ends are useful for DNA cloning because complementary sequence annihilate and they can be joined directly by DNA ligase. Cut and ligate to DNA with echo RI. Here the steps. Cut with echo RI which sticky ends. Here the recombinant DNA. Step 3. Splice or ligate DNA into a cloning vector to create a recombinant DNA molecule. 6 different types of cloning vector. Plasmid cloning vector, Fag cloning vector, Cosmid cloning vector, Shuttle vector, yeast artificial chromosome, bacterial artificial chromosome. Plasmid cloning vector, bacterial plasmid naturally occurring circular double-stranded extra chromosomal DNA element capable of replicating autonomously within a cell. Bacterial plasmid, it's naturally occurring, its shape is circular, double-stranded, extra chromosomal DNA element capable of replicating autosomally within the cell, naturally in the cell. Plasmid vector engineering from bacterial plasmid vector engineered from the bacterial plasmid for use in cloning. Required feature of coli plasmid vector. First, origin sequence required for replication. Selectable trait that enable E. coli that carry the plasmid to be separated from E. coli that don't. Any trait We can notice, we can measure antibiotic resistance, grow cell on the antibiotic. Only those cells with the antibiotic resistance grow in the colon. Unique restriction sites such as that an enzyme cut the plasmid DNA only once. Fragment of DNA cut with the same enzyme can then be inserted into the plasmid restriction site. Simple marker that you or allow us to distinguish plasmids that contain insert from those that don't, like Z, G plus G. Here, BUC19, polylinker restriction enzyme, origin of replication of the sequence, origin, ampicillin resistant gene, origin of the sequence, here, the origin of the sequence, here, multiple cloning site, here, the ampicillin restriction gene, ampicillin restriction gene, is part of the beta galactosidase gene. Multiple cloning site, polylinker restriction site. This is the vector BUC90. Here the plasmid BCU90. Plasmid confer resistance to ampicillin and can make functional beta galactosidase. Here the restriction cut in polylinker, cut with restriction enzyme. DNA insertion in distributed Z gene. Plasmid confer ampicillin resistance but can't make functional beta galactosidase. Fag cloning vector, engineered version of bacteriophage. Central region of beta chromosome, linear is cut with restriction enzyme and digested DNA is inserted. DNA is packaged in fag head to form virus particles. Fag with both ends of lambda chromosome and 20, 30, 37, 50, about 2 kilobase per insert, replicate by infecting E. coli. Phage replicate using E. coli and the lytic cycle, produce large quantities. Like plasmid vector, large number of restriction sites available, useful for large DNA fragment than BUC90. Here, the phage assembly with the release of the protein infection, phage DNA, lytic response, and the lambda chromosome replication, breakdown of the host DNA, replication of the chromosomes. Here enter the lytic cycle, excision of the chromosome, introduce of lytic cycle, chromosome, integration of lambda chromosome into host chromosome, the host DNA, phage DNA, lysogenic response, integration of lambda chromosome into host chromosome, lambda chromosome here, induction of lytic cycle, excision of lambda chromosome, lambda chromosome replication, of the lambda chromosome, assembly of the phage protein. This is the whole lytic cycle. Here the phage DNA, E. coli, EcoRI, not necessary for alpha replication. Arms contain all the genes needed for the replication but are too small for packaging. Here they need to be cloned. Restriction fragment of the different size, suitable size for packaging after ligation with arms. Packaging of DNA in lambda factors using protein and enzyme from lambda infected E. coli. Infection of E. coli cells for lambda replication to clone the DNA. Cosmic clone vector. Sorry, feature of both plasmid and phage cloning vector. It takes both the features of the plasmid and the phage cloning vector. Don't occur naturally. Origin, sequence for E. coli, selectable marker, restriction site, fact, cause site, permit packaging into alpha, lambda fuse, phage, and introduce to E. coli cells. Cloning site, here ampicillin restriction. This is a cosmic site, the origin. Capable of replication in two or more types of hosts, replicating autonomously or integrate into the host genome and replicate when the host replicate. Commonly used for transporting gene from one organism to another, transforming animal and plant cells. This is the shuttle vectors. Shuttle vectors capable of replicating in two or more types of hosts, replicate autonomously or integrate into the host genome and replicate when this host replicate, commonly used for transporting gene from one organism to another, transforming animal and plant. Here, for example, insert firefly luciferase gene into plasmid and transform agrobacteria, grow agrobacterium in large quantities and infect tobacco plant. Yeast artificial chromosome, vector that enable artificial chromosome to be created and cloned into yeast. Yeast telomere at the each end. Telomere is the end of the chromosome. Here's the chromosome, and here the telomere. Yeast chromosome syndrome sequence, selectable marker, amino acid dependence on HR, autonomous replicating sequence for replication, restriction sites for DNA ligation, useful for cloning very large DNA fragment up to 500 kilobytes, useful for very large DNA fragment. Bacterial artificial chromosome vector that enable artificial chromosome to be created and cloned into E. coli, useful for cloning up to 200 kilobase pair, but can be handled like regular bacterial plasmid vector, useful for sequencing large stretches of chromosomal DNA, frequently used in genome sequencing projects. Like other vector, bacterial artificial chromosome contain origin, sequence derived from the E. coli plasmid called factors, multiple cloning site, restriction site, selectable marker, antibiotic resistance. Recombinant DNA libraries, genomic library, collecting of cloned restriction enzyme, digested DNA containing at least one copy of every DNA sequence in the genome. Chromosome library, collecting of cloned restriction enzyme, digested fragment from individual chromosome. Complementary DNA library, collecting of clones of DNA copies made from the single RNA isolate from the cells. Reverse transcriptase, RNA-independent DNA polymerase, synthesize DNA from the DNA template. Complementary DNA libraries reflect what is being expressed in cells. Clones required for complete library can be calculated from the size of the genome and average size of overlapping fragment cut by restriction enzyme. Library should contain many times more clones than the calculated minimum number of clones. Genomic library. 3 ways to make genomic library. Complete digestion at all relevant restriction sites. Produce a large number of short DNA clones. Genes containing 2 or more restriction sites may be cloned in 2 or more places. Mechanical shearing. Produce longer DNA fragments. Ends are not uniform, require enzymatic modification before fragment can be inserted into a cloning vector. Partial digestion. Cut at least frequent restriction site and limit the amount of time the enzyme is active. Result in population of large overlapping fragment. Fragment can be size selected by agarose electrophoresis fragment have sticky end and can be cloned directly. Partial digestion of DNA by restriction enzyme generate a series of overlapping fragments, each with identical 5 GATC sticky ends. Resulting fragment may be inserted into a BAM-HI site of plasmid colony vector. DNA fragment from such digest hybrid site can be created by source C3A, but not by BAM-HI. Is the vector plasmid. Partial digestion with SAU3A results in library of overlapping DNA fragments of various sizes. Screening a genomic library, plasmid or cosmid, plasmid vector containing digested genomic DNA are transformed into E. coli and plated on selected media, ampicillin, for example. Colonies, colonies that grow are replicated onto a membrane. E. coli continues to grow on the membrane. Bacteria are lysed and DNA is denatured. Membrane-bound DNA is next probed with complementary DNA radiolabeled by phosphorus tacky. So, complementary DNA in the group is composed of DNA sequence you are looking for, homologous sequence presumable also found in the library. Unbound probe DNA is washed off the filter. Hybridization of probe DNA is protected by exposure to X-ray or by choluminescence. Pattern is noted for exposure pattern clone on X-ray film. Select clone that test positive and isolate for further analysis. Screening a genomic library. Transport E. coli with a genomic library here made in plasmid vector. Velvet surface sterilized placed on the plate on the selected growth medium colonies grow. Velvet Kenyan cells from original colonies is proceed to selected medium plate will filter on its surface. Replicate plate colonies onto new selected medium plate with membrane filter surface colonies grow on the filter. Filter removed from the culture dish bacterialized DNA nature and bound to the filter. Probe DNA hybridized to DNA on filter. Label probe solution added to the filter in 8 syllable bag. Wash filter free of unbound probe to detect hybridization by auto digraphy of radioactivity label probe of chemiluminescence detection for non-radioactive label probe. Here, chromosomal library screening can be reduced if target gene can be localized to a particular chromosome. Chromosome can be separated by flow cytometry. Condensed chromosomes are stained with fluorescent dye. Chromosome separate based on the level of binding of the dye and are detected with a laser. Complementary DNA library is derived from the mature messenger RNA doesn't include introns. Complementary DNA may contain less information than the coding region. Complementary DNA reflect gene activity of cell at the time. Messenger RNA are isolated, varies from tissue to tissue, and this time messenger RNA degrades quickly after cell death and typically require immediate isolation. Cryoprotectant can be sealed if the immediate freezing is complicated by field work. Creating complementary DNA library, isolate messenger RNA, synthesize complementary DNA, clone complementary DNA. Creating complementary DNA is one isolates messenger RNA, mature eukaryote messenger RNA has a poly A tail and DNA. Messenger RNA is isolated by passing cell lysate over a poly T column composed of oligonucleotides. desoxycyanidinic acid. Poly A tail stick to the oligo-AS and messenger RNAs are retained. All other molecules pass through the code. Creating complementary DNA step 2. Anneal a short oligo primer to poly A tail. Primer is extended by reverse transcriptase 5 to 3, creating messenger RNA DNA hybrid. Messenger RNA is next degraded by RNA H, but leaving small RNA fragments intact to be used as primer. DNA polymerase 1 synthesize new DNA to 5' and remove the RNA primer. DNA ligase connected DNA fragment results a double strand complementary DNA copy of the messenger RNA. Here the poly A tail anneal oligo primer, DT primer, reverse transcriptase, degrade RNA fragment, use a primer for new DNA synthesis. DNA polymerase 1 synthesize new DNA and remove RNA primer. DNA fragment joined by DNA ligase. This is the synthesis of complementary DNA. Creating complementary DNA library. Step 3. How to clone complementary DNA. Complementary DNA has blunt ends, thus need to add restriction site linker or adapter to make them sticky. Use A4 DNA ligase and blunt end ligation to add restriction site linker or adapter to each end of the complementary DNA. Next, digest the linker with the same restriction enzyme used to cleave the vector. Mix complementary DNA with cut vector DNA in the presence of DNA ligase. Transform it into E. coli host for cloning. If complementary DNA has the same restriction site as the linker, complementary DNA will be cloned in pieces. Solution use adapter with single-stranded overhangs that match the restriction site on the vector. Cloning of complementary DNA using BMHI linker. Alternative use adapter, double-stranded DNA, E4 DNA ligase, cleavage of linker with BAM-HI, insertion into vector, cleavage with BAM-HI, vector. Screening of complementary DNA. Complementary DNA libraries are most often used to detect gene or protein, are generated for genes that are transcribed. If you know that the DNA sequence for a protein coding gene you want to find, a homologous DNA probe can be used. If no homologous DNA sequence is available, complementary DNA can be probed with an antibody that recognizes the protein. Expression vector clone complementary DNA is inserted between a promoter and the transcription terminator before it's transformed. The single RNA is transcribed from the complementary DNA and translated. Colonies now expressing protein are transferred to membrane. Membrane is incubated with radioactive label antibody probe that recognize the protein. Non-radioactive chemiluminescent probe also are available. Colonies with bound antibodies leave a dark spot on X-ray film. Screening of complementary library with antibody probe, recombinant DNA plasmid, expression vector, transcription terminator sequence, promoter, and here translation start origin. Transform E. coli with complementary clone in expression vector. Plate on selective growth medium, colonies grow. Transform individual colonies to micro titer dish and grow store. Bring colonies to membrane, filter and grow on medium. Complementary DNA protein product is expressed. Take filter off and lyse cell in situ protein in product of complementary DNA is about to filter. React protein on filter with radioactive antibody. Wash off the unbound antibodies. make photoradiograph and identify the colonies. Screening of complementary DNA library identification of the complementary can be used to isolate and sequence the gene for the protein either with or without any sequence information. Identify and compare homologous sequence in other type of organisms. Quantify amount of messenger RNA synthesized from a gene and measure the level of gene expression. Synthetic oligonucleotide might be used to prove the library in the DNA sequence are available. Degeneracy of the code may require a mix of different oligonucleotides. Degeneracy of the code may require a mix of different oligonucleotides. Complete clone complementary DNA from other organisms showing a high level of homology also can be used as approved heterologous protein. Analysis of restriction enzyme sites. Restriction sites where restriction enzyme cut DNA sequence can be mapped by cutting DNA with selected restriction enzyme, electrophoresing the DNA on the agarose shell, and visualizing the DNA band pattern with acidum bromide. Mapping the position of restriction sites generates a linkage map that can have many applications: forensic, systematic population genetics. DNA is cut with different enzymes, and each DNA enzyme mixture is loaded on the separate lane in the gel. Negatively charged DNA separate by size in the electric field. Smaller fragments move faster than the larger fragment. Fragment pattern produced by gel stained with the acidum bromide is photographed or imaged and processed by software. Distance each band migrate in calibrated with known size standard of 3 cut DNA ladder. Resulting pattern of different numbers and size of fragment cut by different time is interpreted to make restriction site map. Here, the large band, the migration, small band migrate faster than the coli, the band H1. This is the base pair size of the bands with kilopair, colobase pair. Many copies of the colonies, 5 kilobase pair linear DNA fragment. Analyze DNA by restriction enzyme cleavage and agarose shell electrophoresis. Large DNA migrate and short DNA. Construct calibration curve for marker. Here, distance migration, and this is the result, interpretation, and construct the model and conclusion. Application of restriction enzyme site analysis. Analyze gene structure with large. Application of restriction enzyme site analysis. Analyze gene structure with large and fine scale mapping, limited by number of restriction sites in the target sequence and the variety of enzymes that are selected for analysis. Restriction fragment length polymorphism can be used for forensic and phylogenetic analysis. Each individual possesses a different pattern due to a variation in number and the composition of different restriction sites. This explains the DNA sequencing other genotyping methods. Cut DNA can be probed without cloning using the Southern blot, which can be used to determine if the gene shares homologous with the probe and how many copies of the gene might exist. Southern blot, invented by Edward Southern after restriction enzyme cutting and electrophoresis, genomic DNA appear as a continuous smear on agarose shell. The natural DNA in the gel needs alkaline solution. Neutralize gel and place a blotting paper that span a glass plate and dredge into the buffer solution. Place hybridization membrane over the gel and stack paper towel and away on the top of the membrane. Buffer solution is waking up by blotting paper to the paper towel, passing through the gel and transferring DNA to the membrane. Fragments on the filter are arranged the same as on the gel. Saturate the membrane with the labeled probe, radioactive or non-radioactive, and expose to X-ray field. This is a plot component of photo here. Wet paper towel, membrane gel, blotting paper, glass plate, tray with cover. Here, cellular DNA, cut restriction enzyme, restriction fragment of the length determined by the location of the recognized sequence for restriction enzyme. The gel electrophoresis of the fragment transferred to membrane filter by Southern blot technique. DNA fragment transferred exactly as they were arranged in the agarose gel, hybridized with labeled probe. DNA fragments complementary to the probe are visible after autoradiography or chemiluminescence. Chemiluminescence. Here, the agarose gel, here paper towel, membrane filter gel, and the blotting paper and tray containing buffer solution. And we can see it in animation better to understand the steps for making southern blot. It's very important in HIV, confirm the infection with HIV. Northern blot, similar to the southern blot, but used to probe RNA, not DNA, can be used to determine the single RNA size, detect differences in the promoter and terminator size, can be used to determine if a particular gene is expressed, and if so, how much, what tissue type, and when in the life cycle. Expression of C2 gene at different life stage in the frog xenopus leaves